Hello, my name is Jessica and today I'm going to discuss genome editing impacts on pharmaceutical industries. Scientists for years looked for cellular repair mechanisms to manipulate DNA through genome editing. The first genome sequence was completed in 2003 and there was a global collaborative effort that started in 1990, therefore taking 13 years to complete. It cost approximately £2.6 billion. And now, a human genome can be sequenced for only $600. The declining cost in genome sequencing results in more research for the study of diseases. They use DNA sequencing for genetic variations or mutations to show the cause of the disease. Then, the disease-causing change can be deleted, inserted, replaced, or modified depending on the genome editing technology type used. The several topics I'm going to discuss today are what is genome editing, the drug discovery process, pharmacological trends, and entrepreneurial issues, and lastly, ethical considerations in drug discovery. Now, what is genome editing? A genome is an organism's complete set of genetic instructions called DNA. DNA sequencing is the exact order of bases in a strand of DNA. Now, genome editing is a type of genetic engineering in which DNA is inserted, deleted, modified, or replaced in the genome of a living organism. Now, how does it work? Scientists can alter an organism's DNA in 4 ways. These are genome editing technologies. The first one is restriction enzymes, which is a protein produced by bacteria that cleaves DNA at specific sites along the molecule. In the bacterial cell, restriction enzymes cleave foreign DNA, therefore eliminating the infecting organism. Then we have ZFNs, which is zinc finger nucleases, or artificial restriction enzymes generated by fusing a zinc finger DNA binding domain to a DNA cleavage domain. Then we have the talins, which is a transcription activator-like effector nuclease. Are also artificial enzymes but can cut DNA strands at any desired sequence, which makes them an attractive tool for genetic engineering, for example, designer babies in IVF, as well as CRISPR-Cas9, and that is the next one we're going to discuss, because they use programmable nucleases to create double-stranded breaks at the target DNA sites to generate desired DNA modifications. This is also extremely important in diseases. So now we have the drug discovery process. The drug discovery process is when a new potential medicine can be identified. It underpins the entire pharmaceutical industry sectors, including research, formulation, trials, manufacturing, and market access and sales. The minimum it takes is 10 years. The first step is discovery and development. This is beginning with the identifying a target for a drug to act on. It begins with hit and target identification. This is to understand the cause of the disease and know what pathways to use for the drugs. And then we have lead identification, where the compounds are screened in a laboratory and they are testing around 5,000 to 10,000 compounds. This is to see the ability, affect identified target. And then we need to check they don't interfere with any other related targets, which causes more deficiencies or diseases. And then by screening, we discover the perfect drug candidate as well, and the compound structure is modified until we find a complete match. And then we find the step 2, which is preclinical research, which is either in vivo studies, which is in the living, which is animal testing, or vitro studies, which is in the glass. For example, cell experimentation, testing efficacy against target and toxicity, which is we're now testing 250 compounds. And then we have step 4, which is clinical trials. This takes around the longest, which is between 5 and 8 years. And that's when we're in full development. We're testing maximum 5 compounds. And there's 3 phases. Phase 1 is determining the drug side effects. Phase 2 is how the drug is effective. Phase 3 is a dosage and safety in a large population. And then we have stage 4, which is FDA review and approval, and that is the Food and Drug Administration. Therefore, they are reviewing the evidence provided by the pharmaceutical company. And if the drug is effective in stage 3, that's when we submit to regulation for the approval. And the FDA's job is to check the production process, observing all the records, collecting samples, and normally only 75% of submissions are actually approved. And then we have stage 5, which is post-release monitoring, and that's when the FDA is surveilling when the drug's gone to market, the risk assessment, and monitoring long-term effects. Now we have the genome editing pharmacological trends. So pharmaceutical companies approach genome editing for managing or even curing diseases at random and untargeted integration of normal genes into disease-causing defective genomes, which decreases the disease symptoms. So comparing all the genome editing technologies, TALENs are cheaper and faster to produce than ZFNs. ZFN's design and construction are difficult and with a moderate cytotoxicity, and they actually take 1 to 2 weeks for construction, whereas TLNs are only 5 to 7 days. And then CRISPR, which is the most ideal genome editing tool for scientists, because it actually only takes 1 to 3 days, and the cytotoxicity is really low and it's very easy, and it also has unlimited gene mutations, which means the possibilities are endless. Since CRISPR was introduced in 1987, most genome editing research projects will use this tool. Also, it speeds up the drug discovery process because it's faster, it's the easiest and cheapest genome editing mechanism, as I've already mentioned. And while all these genome editing tools have been revolutionised in the pharmaceutical industry, in 2018, a researcher called He Jiankui revealed to the world that he made the first CRISPR genetically edited babies using a technique to modify the specific genes on the embryos to make them resistant to HIV on multiple couples, and he was successful. Now we're going to talk about the ethical considerations in drug discovery, which is weighing out what is right and wrong. This is done by the FDA oversighting the testing in drugs in human and animals. Also, in animal research sites are supervised by the Institutional Animal Care and Use Committee. This is ensuring that the test is done as humanely as possible. Also, the scientists have a duty of care to animals. They also have a duty of care to humans in humans trials, therefore informed consent so they know the risks and implications and significance. Also, justice and equity. There is also talk about the increasing divide between wealthy and poor, becoming an increasing divide. As well as genome editing research involves embryos, and this is many religious and moral objections. And then we have the National Institute of Health. They don't actually fund any gene editing in human embryos due to the ethical implications. As I've previously mentioned, the first genetically modified babies have been successful. Although it was not illegal, He Jiankui broke the action of ethical standard on conducting unproven research on humans. Therefore, he was found guilty of illegal medical practices and sentenced to 3 years in prison. So now we're going to discuss entrepreneurial issues concerning genome editing. Figure 3 shows a simplified gene editing process of a chromosome. In that chromosome, there's a defective DNA strand. And then we have the DNA cutting enzyme cutting out that defective DNA strand and placing in a healthy DNA strand. For example, we have unwanted and unintended changes in human embryos where the DNA enzyme tool actually cuts out the defective DNA strand, but as well as cutting that out, it cuts out extra healthy DNA strands. And then we also have disrupting important genes in target cells. Columbia University found while using the CRISPR gene editing technology to repair a gene in human embryos that caused hereditary blindness, they also found additional unintended changes, eliminating entire chromosome or large sections of DNA. This issue scientists are faced with because the alteration risks are so high and can result into more diseases. Then we have commercial viability. While introducing the world to designer babies, the profit in the market is extremely high because everyone wants their baby to be disease or defective free. So the commercial viability is also extremely high, but including the entire idea of designing your baby, for example, their eyes, their hair colour, their IQ, and also all sorts. But again, the main issue is that for ethical considerations, and it will only also be accessible to the wealthy, and the success rate is also low, even though it has been done. So here we have a Q&A for you all today to test your knowledge learned. Is the DNA strand a single or double helix? What genome editing tool is the cheapest? What is the stage 4 of the drug discovery process? So here we have your answers for the Q&A. The question 1 was double. Question 2 was CRISPR. Question 3 was FDA review and approval. Thank you for listening.